Well, it's great to introduce Joe Perry. He's going to be our first speaker on genetically modified organisms, agriculture, ecology and food, theology and safety. Joe worked at Rothamsted Research Centre for 30 years in the area of quantitative methods applied to ecology. And he's jointly responsible for the design and analysis of UK farm-scale evaluations of GM crops, and currently chair of the Genetically Modified Organisms Expert Panel of the European Food Safety Authority. And I think you've come hot foot from the continent this weekend, haven't you? And if he doesn't get enough of plants at work, he lists as his main hobby gardening, so that's great, really dedicated to the plant world. And it's fantastic to have you, Joe, and we look forward to hearing your talk. Thank you very much.
Thank you, Margot, and thanks to the organisers for inviting me. Okay. Everything's risky. GM crops are no exception. I'm a risk assessor, so if they weren't risky, I'd be out of a job. Some of the risks, 4 of them here, are scientific, and those are the ones that I'll address. The others, equally, and as Christians, some might argue more important, I won't cover. Other more talented speakers than me will cover those. So this is the EFSA GMO panel, as it was last year. 21 experts, independent academics, and I'll try to tell you something about their work. Christians have quite a lot to say in this area, and as Margot said, it's very controversial. I asked someone from a Christian Green organisation what their concern was. These are the concerns that they gave me. Now, some of them are scientific and some aren't. The ones above the line, in general, less scientific, but you can see some of the arguments there. You might call them socio-economic, the ones in blue. Contamination and coexistence, now we're moving maybe midway between science and the business of farming. And in fact, I'm a risk assessor, so I don't deal with those issues. It is the risk managers, so-called, who deal with them. So again, I won't be dealing with those issues in green. But then when we come down below the line, we do come to the kind of issues that we discuss when we're looking at applications to import or grow GM food, feed, and plants. I will throw in a question which we might debate later. I can't see any scientific or ethical reason why GM couldn't be integrated into organic agriculture. Maybe that's something we debate. But most major technologies have got their benefits and their risks. Mobile phones can be used in a beneficial way, in a risky way, explosives also. Interestingly, in other parts of the world, they do look at benefits. We don't in Europe. We assess the risks regardless of the benefits. We also differ in that we assess the risks of the technology. In other words, we assess the risk because it is GM. I put regardless of the trait, that is, whatever the trait is. If it's GM, we assess it. We take account of what the trait is, obviously. Canada does things differently. They assess the trait itself, regardless of the technology. So, for example, some GM crops are what's called herbicide tolerant. That means that you can apply a particular herbicide to them, the farmer can, in order to control their weeds. Now you can develop a herbicide-tolerant crop in a way through genetic modification or conventionally. So Canada will assess the risks in exactly the same way, but we in Europe don't. And we have a directive and a regulation within Europe that really governs the way in which we assess and what we have to look at. I won't dwell on the theological implications because, again, there are people who are much more qualified than I who are going to talk after me, but I will just draw your attention to the fact that I did publish this paper in Science and Christian Belief, which tried to look at them. In fact, there have been many individuals and organisations, especially Christian organisations, that have taken a view, and particularly I think when this issue was last in the news around the late 1990s, there was a lot going on, a lot being published, and again, as Margot said, a wide range of views amongst those contributing. I summarised mainly on the basis of the fact that all technologies can be used for good and for evil, depending on how they're used, and that usually they're fairly broad, and also on the basis that obviously they're not explicitly referred to in the Bible. How could they be? So I argued that growing GM crops does not have consequences that must of necessity be outside God's will. And at that time we were concerned, particularly in the UK, with herbicide-tolerant crops, and there were serious concerns about their effects on wildlife through the great suppression of weeds, and one person's weed is another person's arable plant, and is food and shelter for many insects on which farmland birds depend. So at that time there was quite an emphasis, and that paper has an emphasis, I think, on the herbicide-tolerant aspect. So I'm going to leave that now and just try and give an overview of what we do within Europe. So anytime someone, a company, wants to import a food or a feed or grow a crop, it has to be authorised at the community level, and it has to have a risk assessment regarding its effect on human health, animal health and the environment, looking at potential adverse effects. They may be direct or indirect. So in the case of herbicide tolerance, there's no direct effect of the actual plant itself in the field. The effect on the environment comes from the herbicides that are applied to it in their management. So that's an indirect effect. Effects may be immediate, they may be delayed, or they may be cumulative. So the European Food Safety just is an advisory body of scientists. We advise, the European Union decide. We have 27 member states and they vote. The Commission need a qualified majority to give approval, the Commission being like the civil service of Europe. And although the World Trade Organization rules say that the decision should be taken on science grounds alone, actually the votes are done on a political basis. And because of that, they very rarely get a qualified majority. And that's especially true for cultivation, which is much more sensitive than the import of food and feed. And if they don't get a majority amongst the member states, then the Commission itself have to decide, and they have decided in the past, often after some length of time, and you'll see that in a minute. So here we've got the member states' voting patterns. You can see in red those that vote against, green those that vote for, Some people always seem to vote for, some states always against, so clearly it's not done on science grounds unless there's absolutely no consensus. And there's a wide range from Austria and Hungary, who are in general very cautious, to our own country, which by and large has voted in favour. So as a result of that, we have, as I said, we've got now 39 products approved for food feed import, but only 2 approved for cultivation. Monate 10 maize, which is an insect-resistant maize, and a starch potato, which is not for human consumption, which actually the company has now withdrawn, so won't be grown. So effectively we're back down to 1 product, whereas in other parts of the world, particularly the United States, where and Canada, where they've probably been growing and eating GM food for longest, there's less stringent approval process. And of course, it takes a fair time, even in the United States, for approval. It takes a lot longer in the European Union, and in fact, we are still assessing some applications that were initiated in 2005, I think, for a number of reasons. I mean, we ask questions. Sometimes the companies are very slow in answering, and sometimes I think they have little desire to actually get to the end of the process for one reason or another. So again, that's telling a similar story. Typically, yeah, it would be over maybe 2.5 years that the application is looked at within the organisation I work for, and then it goes to the European Commission, and then there's a further delay there. Okay. Now, what about the scope? I mean, what is it? Well, first of all, food. We know what food is. Feed is the same thing, but for eating by animals. Then there's deliberate release into the environment, which is another risk that we look at. Of course, if you import food and feed, you can also have a release into the environment, because you can have spillage at the docks, spillage of seed. That seed can then germinate, maybe, and so we have to look at whether that seed will then become feral, for example, and set up a population, and what the risks of that might be. And then we have cultivation as well, where if you give permission, you're actually giving permission to a farmer to actually sow the seeds and grow the material. So what we do within EFSA is we do a scientific risk assessment, and we don't look at the socio-economic areas at all. And we typically will look to see whether the GMO is as safe as its conventional counterpart. So we have conventional maize, and so, for example, maize Monate 10, which is a particular variety, which is genetically modified. It has a parent that was used for the modification. So typically we will compare the genetically modified with the nearest, which in this case is its parent, maize. And we do a comparative analysis, so we are assessing safety on a comparative basis. And what kind of expertise is there? Well, we typically split into 3 working groups, and there's about there's meant to be 21, in fact we've only got 19 experts on the panel at the moment, and they would split into looking at the molecular characterisation, that is the genetics, what sequence was actually put into the DNA, and what implications does it have, if any, for safety. We would look at food feed safety, so typically the crop will be grown and then it will be, its composition would be analysed chemically, split down into various variables such as how much ash has it got, how much sodium, how much glycemic acid, how much this, how much that. And then there's a toxicology assessment of allergenicity. We look at its nutrition, does it do what it's meant to do? And then, particularly for cultivation, but also for food feed, but particularly for cultivation, there are a bunch of ecologists who look at environmental risk assessment, looking at things like will there be persistence and invasiveness of this? What will happen when it hybridizes with weedy near relatives? What happens when it hybridizes with crop plants? And what are its effects on other organisms such as bees, beetles, and such like. So there's a range of scientific assessments, and quite a lot of people, as you can see, who contribute to that. And there are also, the member states have their own experts and feed in questions to be considered, and they are working for their own governments of the member states. I'm going to try and give you just a couple of examples of the sort of scientific challenges that we have, because it's much easier, I think, dealing with concrete examples. Now, this example is about the basis of a problem that we have when we look at composition and analysis. In fact, this is, I think, a problem in risk assessment generally. So, we should be aware that when we do an experiment, we usually are testing a null hypothesis. So we put a null hypothesis up to be shot down, if you like. The null hypothesis here would be that there is no difference between a GMO and its comparator, which is not GM. Okay, now who does this? Well, actually it's the applicants, the industry, who submit, as we will see, the dossier of information. And it's quite well known in science that experiments have power, or lack power, which is the ability to detect an effect or a difference if there is one there. There may not be one there. Usually when we look at different things, there are differences, so we do find them. But the ability of the experiment can vary, and it varies depending on how much effort we put into it. Can vary in terms of the replication, in other words, how many times we redo something in order to make sure that we've got a good result. And because of that, society at large, in risk assessment, rightly needs to be assured that the person who's done the experiment has put enough effort into doing it, that if there is a difference there, they're actually going to find it. Because if we do an experiment with maybe just 2 replicates instead of 10 replicates, okay, we may find some difference, but the noise, the variability may mask that. And so this is a common problem, I think, in risk assessment, in that the risk assessor has to be assured that whoever's providing the data has put enough effort in, that their experiment has enough power to detect the difference, if the difference is there. So that's an introduction to this problem. So here we can see means of a particular endpoint, that's a particular variable, won't specify what it is. And we can see the two means and their confidence limits, which describe their variability. And because they're not overlapping, we can see, yeah, we see there is a difference here. Okay. The question is, what does that difference mean? Because we can find a statistical difference, and that may not be biologically relevant. And then the toxicologists have to decide, well, okay, there is a difference in this endpoint, whatever it is, the amount of sodium, for example. What does that mean in terms of food safety? Well, one way in which we can assess that is by what's called the principle of the history of safe use. This goes that, well, we've been eating maize for about 10,000 years, or developing it for about 13,000 years. We believe that conventional maize is safe, and therefore let's look at where the GM sits in relation to all the values that you would get for this particular variable within, and to see whether the GM fits within that range. So that's a different approach from looking at the difference. That's trying to put the difference, if there is one, between the GM and its comparator into context. Now, because of that problem that I alluded to before, of applicants maybe not putting enough effort into doing their experiments, we need, and we've recently developed a different approach, and I'll explain why in a minute. But just to say that commercial varieties, if they have a range, we can quantify that range, we can form a distribution, statistical distribution that is, whose mean and variability we know, and then we can assess the GM on the basis of that variability from the commercial varieties, which we now insist are grown at the same time in these compositional trials. So it's all the reason we are doing this new equivalence test comes back to this problem of risk assessment, because there's a difference between getting a false positive and false negative. And this illustrates the difference. So on the left, you're pregnant, that's a false positive. You're not pregnant, it's a false negative. And I won't dwell too much on the next slide, but the idea is to try and explain to you that it's actually relatively easy to fix this type 1 error, this false positive. That is saying there's a difference when actually there isn't one. That's easy to fix. This is what the consumer is much more concerned with, this type 2 error. That is when there is a difference and we falsely conclude, maybe because the experiment hasn't been replicated well enough, that there isn't a difference. And that's down to the form of the null hypothesis that's used, that's typically used. Typically we say things are the same, that's our null hypothesis, then we try and disprove it. So here there's a problem, because this type 2 error is difficult to set. It's difficult to know exactly how much power there is in that experiment. Whereas the type 1 error, which is the risk to the producer, the applicant, that is easy to set. So the idea that in the new food feed guidelines, which actually have been adopted by the European Commission on last Monday after a delay of about 5 years, that will be hopefully overcome by the need to do not just a test of difference but a test of equivalence. And the test of equivalence turns around the null hypothesis so that now the potential risk for the consumer is easy to set. And the onus really then is on the applicant to do a sufficiently well-resourced experiment so to reduce their own risk of finding false positives. So that's a kind of food safety and statistical example. And I realise that that might be difficult to take in. The next example I hope will be a bit easier. Here we've got the environmental risk assessment for non-target Lepidoptera. What do I mean by that? Well, maize monactin, let's take it as an example. This maize, why was it developed? It was developed to try to reduce the damage to maize caused by moth larvae, pest moths, such as the European corn borer. So pest in the United States, pest also in Europe. So, the idea is that you take a gene from a soil bacterium, which is quite specific in its effects on, relatively specific, its effects. It harms Lepidoptera, it harms butterflies and moths, not, in general, beetles, worms, rodents, us, or anyone like that. Okay, so it harms Lepidoptera, but of course butterflies are Lepidoptera, and it might be harming the pest, that's fine for the farmer, but what about the problem that it will harm non-target Lepidoptera? In other words, butterflies, maybe some of them of conservation concern, and so that's the problem. You've got pollen from the BT maize, which is on the right here. The pollen's the black bits, and the larvae, maybe of non-target Lepidoptera, are feeding maybe on nettle plants, and if the pollen lands on the leaves of the plants, the the butterfly larvae will ingest it as they eat their host plants. So that was the problem. But the difficulty is that's actually quite difficult to measure in the field. In the lab, we've got quite good ways of relating mortality to dose. And we've also got good data that tells us the degree to which pollen density, or the pollen concentration, pollen cloud if you like, declines with distance from the field. And the research involved putting those two things together so that we can actually link mortality with distance without, we've got some field data which is very useful, but it's a way of supplementing that field data. And from that, then, we can assess what the risk is, how big is the risk, how much mortality would be caused, and we can put it in a farming context. And also, so for example, we can recommend risk management. So here on the right, we've got a nature reserve. We could impose a minimum separation distance so that although there might be some small amount of pollen which might reach that nature reserve, we can make sure that it is within a certain boundary that does not have a marked effect. And within, but that might be okay for a nature reserve, but then you'll say, well, how about the edges of fields? And their margins, here on the left, field margins are home to a lot of really useful wildlife, amongst them butterflies. What could we do then? Well, one of the things we could do would be to insist that around the edges of fields we plant strips of non-target genetically modified maize. So the genetically modified maize here, pollen has further to travel and has less effect. And those problems have been recently part of the EFSA opinions. Well, how much evidence is there? Well, this typically is what we would receive for a particular application. It would be a dossier maybe of about 6 typically ring binders full of, I now use that ring binder to store my hymn music in. So it's probably more useful now than it was then. Anyway, so we have a lot of information. Fits on more than one CD. Food feed wise, food feed safety, as I say, we've approved within Europe some 30 applications, and they're being imported. And I am fairly sanguine about food feed safety, but of course the history of safe use for food does not translate well in any case to the environment. We always, whatever we do in farming, we have an effect on the environment, and we have an effect on the biodiversity. I mean, from a physics point of view, you've got the energy from the sun, and we are basically balancing it. We are partitioning that energy, and some of it is going into our farming and our crops, and what's left over goes into biodiversity. But we're always going to have an effect by farming. And so, because of that, the environmental effects of GM crops, we can't assume a history of safe use, because there isn't one. We can look, and at the moment our assessments are based on, is there an effect which is different from that which is conventionally done? So is this system, this GM system, is it having a bigger effect on the environment than a conventional farming? Of course, you can argue that you don't like conventional farming, and that's a different argument, and quite a fair argument. Although there's a lot of work being done to show that we can farm sensitively. An awful lot of research by these organisations and funded by government departments like DEFRA, in order to try to farm, but also make sure that we don't have too great an effect on our farmland. And here I think this is important to us in the UK, because to a certain extent we live in our villages, amongst our farmland, in a way that the United States don't. I mean, they've got their huge national parks, their farmland is less heterogeneous, has less maybe natural biodiversity within it. They lack the small woods, the hedges. So for that reason, I think that that there is still, within the biotechnology industry, there is a reluctance to look holistically at this aspect of pests and farming. In other words, what they will say is, I've got this herbicide. Herbicides are meant to kill weeds. That's what they do. That, in fields, we shouldn't really be concerned about what biodiversity we've got in fields, because we basically want to get rid of weeds so that we've got clean crops. So that I think is a fair description of the attitude of industry. It's not the attitude that we have. We take the view, and Europe actually now takes the view, in a recent directive, European farmers are being told that they should use integrated pest management. Now, I'm not sure to what extent this is going to be imposed by Europe, but it's certainly, we are moving towards use of less pesticide. Europe has withdrawn several pesticides recently. EFSA itself has had an opinion published about neonicotinoids, which has been in the news recently, and their possible effects on bees. So, there are ways of farming sensitively, and that basically is our baseline, if you like. Our baseline is not organic farming, because organic farming is a fairly low percentage of European farming. And this slide questions whether we could use organic farming to feed the world's increasing population without destroying the environment. And the evidence has been hotly debated. This is a recent paper from Nature which has done a meta-analysis, that is an analysis of several analyses, and came to the conclusion that no, yields, as you might expect, I think, from a purely agronomic point of view, would not be as great. So, now let me try and move on to the basis of GM. Here's a field of GMAs. It's part of the UK Farm Scale Evaluation, so we took half fields, and half of it was GM and half of it wasn't. And actually, well, I'm not sure that I could tell, well, I know I couldn't tell the difference if I went into that field if I didn't know which are, and in fact I've forgotten anyway, so I still don't know. So, but it raises the question, I mean, if you go and look at that maize growing there, it looks the same as normal maize. Why do we have this visceral mistrust, taking a gene from one species and placing it in another? This is an old slide, an old idea now, goes back to over 10 years. A colleague of mine, Keith Davies, who's a nematologist, studies nematode pests. He asked himself this question, and he said, well, I think it's the fact that actually when we think of a species, we are still thinking in the way that Plato and Aristotle thought. We have this idea of a species as an unchangeable type with some ideal form. Whereas actually Darwin talked us about the uniqueness of an individual, which is not an unchristian concept, and thought of the species as a statistical abstraction. So, in evolutionary terms, species do not stay the same forever. And Keith said, well, actually, I think, despite the fact Darwin is now well over 100 years old, he thinks, well, we're still actually thinking in this way, in the old Platonic way. It's quite an interesting idea. And of course, it's not just the Greeks that focused on species, but the Old Testament too. So the question here is, how many of our genes, I mean, have we got any genes in common with things like bananas and fruit flies and zebrafish? And maybe people would find it less horrifying to take a gene from a zebrafish and put it into a tomato if they knew how many genes in general we have in common. I've got more than 99% in common with my dear daughter Sophie, but 60% in common with a banana. And so on. Future challenges. Biotechnology hasn't stayed still, and particularly transgenesis is just one method. Transgenesis by taking a gene from one species transferring it to another species. But there are all sorts of other methods that are now being worked on in research labs and starting to be used. Not yet, but they will come into commercial production. And the question is, how are we going to regulate them? Are we going to use the same regulations for those as we have for transgenesis? Can we learn from what we've already done? Cisgenesis, for example, is where we do genetic modification, but we don't take it at the top. We've taken maybe a gene from a soil bacteria and put it into an apple, for example, just as an example, to cure some virus disease on the left, and we come up with a transgenic apple on the right that doesn't have the disease. Well, we could do this by conventional breeding also. That's the middle row. We can also do it by cisgenesis, by looking at this wild apple, wild relative, and taking the gene from the wild apple that is responsible for resistance to that disease and putting it into an apple, commercial apple, getting another apple, which is genetically modified, but through cisgenesis. Should that be regulated in the same way? Is it as horrifying to people? Those are the kind of challenges that we've got. We've also got the challenges of new traits. The traits that so far have been used, I've mentioned herbicide tolerance, I've mentioned insect resistance. They account for maybe 98% probably of those GM crops that are grown worldwide are one or other of those. But there are more on the way. We've recently assessed nutritionally enhanced soybean, which has maybe better kind of fats in terms of human nutrition. It creates its own problems. We don't have a simple comparator anymore. We've got salt-tolerant and drought-tolerant crops which are being developed at the moment by industry. And again, there's going to be problems in assessment, lack of a simple comparator in the environment where the crop will be grown, and possible problems that there will be, that we will need to assess a selective advantage if that gene for, say, drought tolerance was to be passed to a weedy near relative, then that weed would acquire the ability to withstand drought and might become weedier. So those are the kind of issues that we look at, we need to be careful of. GM animals, we are just about to, in April, I hope, adopt our guidance, several hundred pages of guidance for how industry should assess the environmental risks of GM animals. So we've got some here, the EnviroPig GM insects, which are modified to be sterile and released to try to control natural populations that spread malaria by reducing those populations down to very low levels, and the well-known GM salmon, which is at the moment going through the stages of risk assessment, and some people feel not very stringent risk assessment in the USA. And I will end by just saying, further reading, if you want to look into this, you can go on our website, the EFSA website. There's an awful lot of material there, and increasingly EFSA realises, because we have been criticised greatly in the past, it's a controversial subject, so there will always be critics, and we have been criticised for not making our opinions transparent, not making it obvious why we've come to the conclusions we have come to. And actually, it's uncomfortable being criticised, but I think necessary for this transparency, and increasingly, with its new executive director, who's been in post about 5 years, EFSA is becoming more and more transparent. We've now put the data for one of the applications on our website. The application that's highly controversial, that was approved, that has been quite controversial recently, for NK603 maize NK603. You can actually, if you want, download all the data and see exactly what it is that we base the assessment on, despite the fact that our executive director has been threatened with legal action. I think it's a good thing. I would like the data to go, all data to go on the website. There are legal and there are other difficulties about that, but I think we are moving towards transparency, and that's a good thing. And within the UK, there are two government committees of scientists that look at these issues. One deals with the environment, the Acre Committee. Again, there's a good website for that. And the other deals with food safety. So I really look forward to answering questions. Thank you.
Thank you. Thank you very much, Joe. That's certainly taken me into an area I haven't had a lot of understanding of before, and thank you very much for presenting that. Have we got some questions? And do briefly say who you are when you ask a question.
Hello, my name is Rachel. I'm an agriculture student, and I was just wondering, you may have mentioned it already, who conducts the compositional analysis?
The applicants conduct the compositional analysis. Usually, they will do field trials, and they now are doing, since our guidance was issued on this about 18 months ago, and the EU, as I said, has now adopted that guidance, so it's now actually a legal requirement. So they will do the compositional trials according to quite well laid out protocols. So, you know, the number of sites, the number of varieties that they will use. They do the trials, and usually they would ship off the material to some lab to be analysed, and then get the results back, and then they will put the results as part of their dossier. So when we assess things, of course, we do depend to a large extent on the information from the applicants in the dossiers. We also, of course, look at scientific literature. And there's an awful lot of material in the scientific literature. So I feel fairly confident that the data that they supply is for food safety is fairly good. The data for environmental, I have confidence that it is not false data, but it is often of an unbelievably poor quality that would probably not even pass muster as a BSc dissertation, so. Well, it can be. I don't want to exaggerate, I mean, but we often have to ask supplementary questions.
Another question.
Chair Lavelle. Could you say something about the so-called doctrine of substantial equivalence? And then perhaps you could go on to say how we know what kind of illnesses could be stemming from GMO when nobody knows exactly what they're eating.
I did mention the doctrine of substantial equivalence, and that is, how do we assess a GM food? If it were a chemical, we could feed it to rodents in high doses. One of the problems with food is that you can't easily increase the dose when you give a rat food in a feeding study. So in other words, there is a technical challenge in producing food for a rat feeding study to look at acute toxic effects, but they are done and they are assessed, and in fact from Monday those 90-day studies are going to be mandatory. I'm not sure how sensitive they would be anyway, but toxicologically I think that they're fairly sound and they've got the confidence of the academic community. In terms of diseases like cancer, what we would do, we would look at these, the particular modification. We'd look to see what proteins are expressed. The toxicologists would look at that. We would do the compositional analysis as well to look for unintended effects. So the basis of substantial equivalence is to say, well, how are we going to look at food safety? And the way that it is generally accepted, and I realise from your question that there are critics of this and criticisms have been made, but I think it's generally accepted that the best way of looking for food safety is to compare, as I tried to say. We've got varieties with a history of safe use, so we look for differences between the GM and that variety. Well, first of all, look for unintended effects. What happened that was unusual? If we find them, EFSA would say, then we would, yes, recommend a 90-day rat feeding study. We have no reason in most of the GMs that we have looked at, I'm not saying this will always be the case, the GMs that we've looked at so far with the traits of BT and HT that we've looked at, there's no reason for us to think that they would be carcinogenic. If they were, there is an argument that has been made for long-term studies. If they were, I believe that we would have indications through the 90-day feeding studies. Admittedly, critics have said, well, the 90-day feeding studies only take you, 1/8 of the way through a rat's lifetime. And people like Gilles-Eric Seralini have said you need to do 2-year studies, which would take us to the end of a rat's lifetime and be far more likely to find any effects, were they there. I can only say that for many years we have been aware of that criticism and have not thought it's necessary to do those studies. There are good reasons why you might not want to do them. One would be the sacrifice of rats. Another would be whether the technical difficulty and the expectation, would they actually tell you anything? I'm also aware, of course, that Seralini has been in the news and is a well-known long-term critic of GM, and he has come up with results which he says demonstrate that GM NK603 can give rise to greater tumours, greater number of tumours. I can only say that we and member states, that is not EFSA but actual member state authorities, including Austria, France, Germany, across Europe, have looked at that study and don't believe that we can conclude anything from it.
Thank you.
Watch this space. Oh, well, I should explain, because the study was conducted in such a poor way, and scientifically it was of very, very dubious quality.
Well, my question follows on immediately from that. John Oliver, I used to talk on these things when I was Bishop of Hereford in the House of Lords. And I went 18 months ago to a conference at Aberystwyth that I was, in which one of the speakers was Michael Antonio, who was a molecular geneticist from Guy's Hospital in London. And he has done 2-year research on rodents, and he has noticed cumulative toxicity. Now, I approach this from a basically pro-GM point of view, but this is the one piece of evidence that has really seriously worried me. I don't hear it discussed. You just mentioned it in response to the previous question. You said EFSA did not think it important to conduct a 2-year study on
Well,
rodents.
we don't think it's
And if you extrapolate
we
the
don't.
effect on a rodent over 2 years, rodents are surrogates of humans, you are looking at 20 or 50 years in human terms with the possibility of cumulative toxicity. Why do you not think that is really important? And that has to be nailed.
Can I distinguish between
It's only just a
toxicity?
minute or so.
Okay. I'd like to distinguish between toxicity, which I think we can study very easily and do, and carcinogenicity. Michael Antonio has not done any rat feeding studies himself. He has co-authored with Seralini, it's part of the same group. We don't think it's necessary because there is nothing that nothing in the makeup or the genetic modification that would lead us to believe that there is likelihood of carcinogenicity. There's no evidence from the scientific literature, and the 2-year studies, as I've said, are fraught with technical difficulties of actually doing them. So we could do a 2-year study, and I believe that the EU might even fund a 2-year study because of the Seralini work. It will be very difficult, I think, to design the protocol for such a study. EFSA is already talking about, if the European Commission say they want to do this, then EFSA will set up a working group to try to develop a protocol which addresses the problems that I've outlined. Mainly of how do you feed rats for 2 years on something they don't normally eat and still get a result which you can believe. That's a real challenge, and I think it will probably take us about 18 months to 2 years to come up with sufficiently good guidance that will stand up. And then it would take about a year to grow the amount of material you would need for a 2-year rat feeding study, and then you would have 2 years. So the result will not come quickly. We don't believe that there is a need for it, but the result may come in about 6 or 7 years. And of course, that will only be on one crop, one event. It may not satisfy those people who are concerned about this.
Thank you very much indeed, Joe. It's been extremely